Hi everyone, welcome to class today. I hope you all guys are having a wonderful day, and I hope everything is going on well. Today's class is about microscopy, and then what we are going to learn, the whole objective is first to match organisms to their size ranges and appreciate how a microscope enable us to be able to see microorganisms. And the second objective is to learn and define three properties of an effective microscopy. And then to distinguish between light and electronic microscope. And then lastly, to identify different staining categories and when we have to use them. So in microbiology, size really matters. And to be able to tell microbes apart, we have categories for them. So one of the category is microscope organisms. So basically, these are organisms which are bigger and it's easier to see them with your naked eye. They usually form within meters and centimeters. So an example include a helminth, some fungi, and some algae. And as you can see from this image up here, this scientist is able to look at, for example, louse without the microscope. And then we have the second category, which is microorganism. So these are smaller, you can see them with your naked eye, and they usually form within the range of millimeters to nanometers. So a prime example is protozoa, and they are protozoa, fungi, or algae. They are usually between 3 micrometers to 1 millimeters. And you have bacteria, which is between 1 micrometer to 10 micrometers. And then you have viruses, which are 20 to 400 nanometers. We have prions, which are less than 10. And as you can see from this image here, as we can all appreciate, when you have microorganisms, you can see with your naked eye, but you have the micro ones, you can't. So you need a microscope to enable you to be able to see. And then as they get smaller, somewhere around 20 nanometers, and it keep getting more smaller, you need something like an electronic scanning microscope, an electronic microscope for you to be able to see it. So within a microscope, we actually have three properties of the effective microscope. And to be able to see a microbe clearly, you need three things, which is magnification, making things bigger, resolution, making things sharper, and contrast, making it stand out. Without all three, you just have a blurry and invisible image. So we first tackle magnification. So magnification occurs in two phases. You have an objective lens, which forms the raw image, and then you have an ocular lens that magnifies into a virtual image. So as you can see, if you have an image, all you have to do is we mostly put the object between the lens and then the light source. The light travels through the lens and then it gets refracted or bends. So depending on the size and curvature of your lens, it either makes it more bigger, um, it, you either get to kind of see your object bigger or smaller based on the enlargement of your optical replica. So basically, based on the size of the lens or the curvature, you get to see things bigger or smaller. And one prime example is this, as you can see, the animal actually in the ladybug actually looks smaller, but when you have a lens, it makes it like more bigger for you to see. So basically, on the microscope, this is how you kind of see things. This is you, this is you have a brain and eye, and basically this is your microscope. So basically you put your specimen on the stage here, and then you have your light source to turn it on. Light travels through here, through the lens, through the specimen, and through the objective lens, as we can see from here. And then from through your specimen basically refracts or deflects some of the light coming from the light source, some of the light rays coming from the light source, and then the ones that get bounced or diffracted goes into this objective lens, and then you can be able to see. So basically, a raw image is formed here on the platform here, but your eyes basically see the virtual image. So to be able to tell to find the total magnification power of what you're actually seeing, basically we have the ocular lens, which is this, and we have the magnification lens. All you have to do is multiply the ocular length by the objective length. So for example, if your ocular is 10x and you are using the 40x objective, all you have to basically what you are seeing is you are seeing the object 400 times. So here we are looking at something which is like 40, we are looking at the specimen through the 40x objective, and then our ocular lens is 10. So all we have to do is 40 times 10, making it 400 times bigger. The same way if you have like a 4x scanning objective and your ocular lens is 10, you just have to multiply 10 by 4, making 40. So you are seeing it 40 times bigger. For example, in the case where like you want to see more things like more bigger, or what you're seeing is extremely smaller, so you need to make it like really really big, then you end up using like 100x objective, which mostly comes, you have to use an immersion oil with it, and basically if you multiply it by 10, it's like 1000x, so it means you are seeing things like 10,000 times of total magnification. So next thing we talk about is resolution, basically the sharpness. So resolution is the ability to distinguish two adjacent objects as separate. And if your resolution is poor, for example, two bacteria might look like one big one. And for technical details too, so for example, resolution is limited by wavelength of light. So you have shorter wavelength provide better resolution than the longer ones. So here, let's focus on this image. These are basically two same identical images, but this one is fuzzy and this one is sharp. So with the power of resolution, we can make a fuzzy image into a sharp image for us to be able to see. Same example is this one, fuzzy, this one sharp. So as I was saying earlier on, we have resolution power. So basically, resolution power helps you to be able to make things more clearer, as I was explaining before from the previous slide. So here you have two bacteria. An example, we have two bacteria here. They're actually two as in their original appearance here. But if the resolution power is bad, or basically if you can't see clearly, they appear like it's basic these two appear as if they are one, and you can't actually see them. And one prime example is this. These look fuzzy as if there's no kind of cellular structures within this cell, but in original or in the reality, they're actually structures. So basically, resolution helps you separate everything, both from the fuzzy state to more clearer state. And then we have contrast. So basically, contrast refers to the degree of bending light as it undergoes. Basically, it helps to pass from one medium to the other. So basically, when you contrast something, it helps you tell the difference between the object and the background. So one prime example is this. You have this image where everything looks more like darker. When the contrast is set really well, you can tell this is all snow and this is actually the background. And one example also equals this, where you have a microorganism where it basically looks the same as the background from here, but when you use contrast, you can tell the microorganism from its background. So to measure contrast is basically measured by quality called refractive index, basically judged by the degree of refraction. So higher refractive index or more light bending gives greater contrast, and too much of light also reduces contrast, which is basically rotated from the iris diaphragm. So as we can see this, I think we've all done this before back in school, where you kind of put a glass over there, you put a straw in it, and you can look at from the side and see how the water bends the straw. Basically, that's how contrast works. So light rays goes through water and air both have different refractive index, and then light rays get bent within the water. And that's how you kind of see that the straw has bent over here. So now let's practice. Question 1. So now you are observing a specimen using a 40x objective on a light microscope in the lab. And then post-lab directions say to include the total magnification in your figure legend. What will you record? And remember that we assume that our objective we always assume that our ocular lens is 10x. So basically all you have to do is multiply 10 by 40, give you 100 times of actual record total magnification. And number 2, now you look back at the system and notice that you cannot seem to get a good view. Specifically, the image is hard to discern from the background. Which principle of microscopy will you use? Yeah, so that should be contrast, because you need to be able to tell the object from the background. And then your specimen slide is missing and you see a single cell organism moving around with flagellum, and the size was measured around 300 micrometers. Which category of microbes will you likely think it falls under? This one, I feel like I shouldn't say it. I should ask about it again in class. But yeah, the answer is protozoa. So now let's do comparison of different microscopes. So now we have 4 kinds of microscopes. We have the light microscope, which visually have subcategories like bright field, dark field, phase contrast, or differential microscope, which is like DIC. And then the step number 2 is fluorescent microscope, basically for measuring or staining of organisms with fluorescent dyes. And we have confocal microscopes. We also have electron microscope. This is usually used to see like really tiny particles, so like viruses or prions. You definitely have to use electron microscope. Don't worry, I'm not gonna like ask you during the exam to kind of like name the 4 subtypes, but you just have to have an idea of what they are. So now let's take a good look at them and then discuss. So first one, what we are going to discuss is the light microscope. And basically you have to, it uses just visible light as source of illumination. You don't need a different source of light. And then here we have the bright field. And as you can see, it just stains, it lets you see the organism as it is. This is an example of paramecium, and we can see from here. And we have the dark field. And then basically you can see that everything else is dark except the paramecium itself for you to be able to see. And over here is a clear example of a bright field, where like you see the organism, and we like, for example, we can see this organism, we can see the cells, we can see what is within it, but basically everything is so bright in such a way the background is a bit brighter too. And then we have the phase contrast. So phase contrast actually emphasizes on cell structures by slight differences in density. So as you can see, the same paramecium, this time around you can see some of the cell structures in it. And then you can clearly see the background help distinguish between it. And then it contains devices that transform the subtle light waves, changing it into changes into the light intensity. Then lastly is the differential interference microscope. Basically, what it does is it's similar to phase contrast, but it just manipulates light to create a well-defined image, and it just appears as 3D. So you realize the same organism which look flat here appears as 3D here. And then we move to fluorescent microscope. So fluorescent microscope basically uses UV as light source, and then this UV basically reflects or stain. Sorry, let me take it again. It just uses UV as the light source, so you are able to see this using these cells or cellular structures using fluorescent dye. So UV hit on the fluorescent dye just refract the UV, and then you were able to see. So this is used for diagnosis and distinguish between cell type and different part of the cells. And then we have the confocal microscope. So it's usually used for 3D viewing. So let's take this. Imagine this is the flat cell that you are seeing, and then you want to know the different we want to image or see them in a 3D, or just image them in different layers of the cell. And basically, for example, this flat cell, as we are seeing here, using a confocal microscope, you can clearly see this, like it's naturally layers, as you can see from here. It's like gives you that 3D shapes, get to see everything that's within it. That's what we use a confocal microscope for. And then we have the electron microscope. So it employs electron beams, which helps in the formation of the image of the specimen. So let's take a quite an example of it. So to be able to actually see this kind of images, mostly samples are slide very, very thin, and they are coated on metals to increase the contrast. So one example is like coronavirus, and I'm sure we all know viruses you can't see with your eyes unless confocal. So as you can see, this specimen itself, or let's focus only on coronavirus. So here, yeah, like the virus, you can see like they are really, really small, and the background can be tell. You can tell the virus itself from the background. The same thing also applies to this one and the other specimens, as we can see from here. So that's how powerful the confocal microscope is. And sorry, the electronic microscope is, and it also helps in magnification or making it possible to see cellular structures. One example is this one. As you can see, this particular cell, you can see the cellular structures within it. And clearly you can see the nucleus in that organism. So now let's practice. So we have, which microscope will we use to image, and which microscope will we use to image a human epithelial cell? So this is the questions and this is the answers. So which microscope will you use to image a human epithelial cell? Yeah, and then the answer is light microscope, because you don't really need this time around, you don't really need, you just need to tell the cell from the background. So you just need a light microscope. What about the Escherichia bacteria? Think here. This one, we actually saw this bacteria in the beginning. Basically, you also need a light microscope for that. How about three, Zika virus? Yeah, I mean, this is obvious because it's a virus. You definitely need an electron microscope. What about E. coli expressing GFP? Just it's expressing fluorescence. So by the name fluorescence, you need a fluorescent microscope. What about rabies virus? Yeah, you will need an electronic microscope. And what about 3D structure of biofilm? Yes, we need a confocal. By the name 3D itself, it gives us a clear view. We need confocal. What about internal structures of a bacteria? Yes. So to be able to view this, I think we will need an electronic microscope, because bacteria is already small, but we need the internal structures itself. So it means we have to magnify it more. What about mycoplasma bacteria? These are extremely smaller bacteria too, so we also need an electron microscope. What about protozoa in a pond water? This, we think we can clearly tell this apart using a light microscope, because you can just see them in the pond water itself. So now let's move on. To be able to look at microscopes, I'm sorry, to be able to look at microorganisms on the microscope, you need to first prepare your sample or prepare the microorganism to make it possible for you to put it on a slide and see it. So the method of specimen preparation or just processing this whole specimen to be able to see depends on the condition of the specimen, which is number one. Whether you want the condition of the specimen basically means whether you're gonna image them while the organisms are alive or it have to be preserved. Or is it like pathogenic? Is it non-pathogenic? What type of species do you have? And then also the aim of the observer. Are you trying to observe the cellular structures or are you trying to observe the extracellular matrix or the file like any structures outside of the cell? Or are you trying to kind of stain them using fluorescence and then tell them apart? And the third one is the type of microscope which are available. So, for example, you want to see, um, let's say you want to tell apart different cellular subcompartment of an organism, but you don't have fluorescent dye to stain them apart. So it means you don't have access to a fluorescent microscope. Then you can employ a different microscope that will help you to tell the different cell compartment. So you can use an electronic microscope for this if you don't have a fluorescent microscope. So yeah, so those are the three conditions. So the type of specimens you have, the aims of your observer, and the microscope you have available. So talking of the first point, which is preparing them the sample, cells suspensions. So we have a different type of preparation of cell preparation for microscopy. And first one is the cell mount. So sorry, the wet mount. So the wet mount includes using a cell suspension in a suitable fluid or water bath. And then basically what it does is that the fluid maintains visibility, provide medium for movement, and then provide true assessment of size, shape, and arrangement color. So as you can guess, this wet mount is mostly more applicable to when you want like the organism to be alive to be able to see it. And then we have smears. So smears are basically trying to fix trying to look at fixed or non-living organism. So by the name smear, it means to spread a thin film made from liquid suspension of cells on a slide. So as we can see from this image, you just made a, a liquid suspension and you just smear or just, you know, scratch, just basically by the name, smear them on it, then you'll be able to see. So basically, either allow it the slide to air dry, and then here, like you allow it to air dry, and then you hit fix the sample on the slide. And then basically the whole purpose of fixing is to kill the cells, and then you kind of secure the specimen on the slide and preserve all the cellular components in a natural state with minimal distortion. So that's the whole goal of the fixing. And you can also fix with either alcohol or paraffin, depending on the goal of what you want to do. So why do we need to stain these cells? So you stain the cells in order to kind of see what you need to see. Are you trying to see the cellular structures or are you trying to study every single part of the cell? So to be able to stain these cells, you will need dyes to do this job. And then these dyes that we use to stain this um to stain these cells are categorized into two. We have the basic, which is basically cationic or possibly positively charged, and they stain this stains the specimen itself. And then an example includes crystal violet, methylene blue, and the list goes on and on. And we have the acidic or ionic. Basically, these are negative stains, and it stains the slide or the background. So there are three categories of stains. We have the simple stain, differential stain, and then the special stains. Should in case we come to the lab, you will be able to see all of these things in person on the microscope. So don't feel bored that I'm describing a lot of things and you just don't get to see it. You'll get to see it in lab in person. So before we move to, so now we know that we stain dyes. We use the dye to stain, and it's either you are using the basic, which is to stain the organism, or you are using the acidic, which is to stain the background of the slide, so that you'll be able to see. So the first thing we tackle is the simple stain. So simple stain, basically, you are staining only one stain or one dye, and we have majority of the dyes. Majority of dyes are like simple stain or basic dyes, and some of them are acidic, but most of them are basic. So one prime example of the basic dye. So the basic dye, we've already learned that the basic dye stains the sample itself, whilst the acidic dye stains the background. So example of the basic dye include crystal violet. Here you can see it just stains the microbe itself, and methylene blue stain the microbe itself too. We have saffron in the one in red also stain the microorganism. But this is an example of an acidic single dye or a single stain. As you can see, necrosin is actually staining the background or the slide itself, but not the organism. So that's how you can tell simple stains or simple dyes apart, whether they are basic or acidic. So basic stains the organism itself, while the acidic stains the background. And then we have the differential stains. So differential stain, basically you're trying to stain two type of organism, and you use two different color stains for this. Usually complex and involve multiple steps. And then some types of these differential stains include gram stain. So gram stain is when you try to stain for like a gram positive bacteria from a gram negative bacteria. I'll explain as in the next few slides. And we also have acid fast stains, which is used to also stain an acid fast bacteria from that of the non-acid fast bacteria. And we have the endospore stain. So basically, you use this to stain for bacteria or fungi or any other thing that have endospores on their membranes. Sorry, that forms endospores. That forms endospores. So let's talk of gram stain. So gram staining, it was discovered by Hans Christian Gram in 1984. And it basically is just a universal way of diagnosing or staining bacteria, diagnosing staining technique for bacteria. Basically, you are trying to differentiate the gram positive, which are the crystal violet or purple, as you can see from here, from the gram negative, which are saffronin or pink bacteria. So then imagine you have a pool of bacteria, you're trying to stain which one is gram positive, which one is gram negative. It's just easy. You can use the Hans gram stains for that. And then we have the acid fast stain, and basically it's used to differentiate acid fast bacteria from non-acid fast, as I've explained earlier. So the acid fast bacteria stains pink. So as you can see, these are the acid fast bacteria. It just basically stains pink or carbazole fusion. And then we also have, this is mostly used in staining of like agents of tuberculosis and leprosy. And then we have the non-fast acid stain bacteria, which appear as methylene blue. So these are the non-fast acid staining, and then the pink ones are the fast acid staining bacteria. And we have the endospore stain, basically to distinguish endospores from vegetative cells. So as we can see in this slide, the endospore staining are showing the ones which are like red in color. These ones, hopefully it doesn't confuse you with my pointer, but these red ones are like the endospore ones. And then the vegetative cells are the blue ones, as you can see from here. And then lastly, we have the special category or the special stains. And these special categories basically pinpoint particular structures or cell structure. So types include the capsule stain, flagella stain, the one I was talking of earlier on, and we have the flat body or type of inclusion stain. So one example of the capsule stain is this one, the India ink capsule stain. And this is used for staining Cryptococcus nymphomans. So as we can see, it's basically staining the whole capsule of the bacteria, and it's basically, you can see it's just highlighting the membrane of the bacteria from here. And then in this figure, we have a prime example of the flagella stain of the Proteus vulgaris. Basically, a stain which is used to build up flagella. So as you can see, it's basically staining a flagella of the bacteria, as you can see from here, compared to these ones which don't have any flagella on them. So let's practice. What is the difference between a simple and a differential stain? So a simple stain uses one dye and stain all organisms and parts, while the differential stain uses two different colors staining to clearly differentiate the contrast cell types or the cell part of the organism. So let me take it again. A simple stain uses just one dye to either stain the organism or the background of that organism. Whilst the differential stain is basically using two different colors of stains to clearly contrast cell types or the cell part. And then let's go to question number 2. So basic dyes are, we can try and choose an answer from them. And then the correct answer is D. So basic dyes are attracted to the surface of a bacterial cell. So with that being said, this will be the end of the lecture for today. Please remember that we have in class on Friday afternoon. In class on Friday afternoon. Please come for lectures on Friday afternoon. We have an in class. In case you have any questions, you can send me an email or you can come in class and ask me. And also remember to do both pre-class lecture assignments that you have to do and also post lab assignments. Always go to module and connect to do that. And also register for the connect so that you have access to all the assignment and you can do it. And also always check on your modules and read before you come to both lab and lecture. Have a nice day. And bye bye. Oh, okay. And I also.
Hi, I'm Gemini. We're about to go live, where you can.
And also, sorry for that. And also remember that the lab courses are actually two. So we have the morning one and the evening one. So always try and know which one you belong to and which instructor which professor you have to send an email to. Okay. Have a nice day. Bye. Okay.